So, what we saw when we did the experiments with these plants, these experiments were only in normal water, and this we will compare with deuterium depleted conditions. So if we put plants into normal water, they will start and switch on the light, they will start to photosynthesize, and they will increase the pH, and this pH increase is associated with a concomitant decrease in the carbon dioxide concentration, and the reverse process applies to when we put the plants in the dark. So when we did the same experiment in low deuterium, 87 ppm deuterium concentration, we, instead of seeing this gradual increase in the extracellular pH, we saw a gradual sudden drop followed by a gradual recovery. And when we did the associated measurements on carbon dioxide concentration, we saw a slight decrease in the carbon dioxide uptake from the water, because this is the uptake due to the photosynthesis. And in deuterium depleted water, this uptake in the first sort of half an hour, it was decreased, but after that the rate of the uptake was similar to that of the control. And when we did the experiment in the dark, so that the plants could only respire, then the normal plants exhibited a decrease in the pH, which was a little bit accelerated in low deuterium water, but the acceleration also happened in the first half an hour. And the associated carbon dioxide measurements showed that this decrease in the pH correlated well with an increase in carbon dioxide content of the water, presumably because of the stimulated respiration. So from these studies we have seen that in the first couple of minutes, or in the first half an hour, when the plants encounter low deuterium conditions, they show a attenuation in the photosynthesis rate and a stimulation of the respiration rate. However, these two actions are not enough to account for the huge acidification, which, as you remember, we could see in the first half an hour when we measured only the pH in the presence of light. So there could be or might be something else going on, which might be an additional factor in causing this significant external acidification. And to find it out, we suggested that might be the membrane potential could be an important factor. As we know well, all living cells develop voltage across the plasma membrane, and this membrane potential is due to different factors. For example, the unequal distribution of the ions across the plasma membrane and the unequal permeability of the ions across the plasma membrane, and the presence of negatively charged proteins in the cytoplasm, and the presence of electrogenic pumps in the membrane, which contribute to this voltage. For example, the animal cells can be schematically simplified with this model that they contain relatively high concentration of negatively charged proteins in the cytoplasm, and this is roughly compensated in molar terms by sodium ions in the external medium. And if the plasma membrane would be impermeable to sodium ions too, as it is impermeable to the proteins, there would be no problem. But unfortunately, this plasma membrane lets some sodium ions into the cells, into the cell interior, and therefore the animal cells have a sodium-potassium pump in their membrane, which compensates for this effect. Otherwise, the cells could not maintain their normal volume. So the animal cells have a sodium-potassium ATPase, which pumps 3 sodium out and 2 potassium in at the same cycle of the functioning of the pump. Therefore, it contributes to the existing membrane potential. Plant cells do not have this pump in their plasma membrane. They have this proton ATPase, and they also have cell wall. And it is this proton ATPase which contributes to the membrane potential in plants. And this membrane potential traditionally can be measured with electrophysiological techniques. So if we take 2 electrodes and put or introduce one of them into a living cell, we can measure current, and the source of the current is in the membrane, because the membrane is the part which separates the charges, and the membrane is the part which separates the 2 aqueous solutions. So the membrane is the source of the voltage, which is the important part in maintaining this current. And the interior of the cell is the cathode, and that has important implications, because nowadays, fortunately, we can use alternative methods to measure membrane potential. We don't need to impale electrodes into the cells. It would be extremely hard to do it with plant cells, because they have a relatively rigid cell walls. But we possess a range of fluorescent probes which can be of help, because they can penetrate into the living cell according to the actual membrane potential, according to the actual voltage. And one of these dyes, which is a fluorescent dyes, a potential sensitive fluorescent dyes, can be seen in this picture. And this is actually a lipophil cation. And since it is a cation, it is relatively easily taken up by the cells relative to the membrane potential. So the higher the membrane potential, the higher amount of this dye is taken up during a given period of time. So that's the principle of the measurement. If the membrane potential is low, there is only a little amount of dye which is taken up. If it is higher, we can get more dye in the cells, and if the potential is even higher, we will have even higher amount of dye in the cells. And since it is a fluorescent dye, we can measure the fluorescence which comes from the cells, and therefore we can estimate how big the membrane potential is. So we did this experiment with low D water, and what we saw was that in the first half of the experiment, in the light, the plant cells took up a huge quantity of this dye, and after that the rate of the dye taken up was relatively similar to that of the control. And that can explain, at least partly, why we observed this relatively significant external acidification. So, in conclusion, we can say that the plant cells are capable of sensing the sudden change in the deuterium concentration of the ambient solution.